Hi, folks. All right, so it is October 26th, Wednesday, October 26th. We have our lecture number 7 right over here. I thought we had a really terrific class last week, so thank you all for, really just giving me 100% on all of your efforts. Just housekeeping note, the area on Blackboard for assignment 6 has been created, so you can go ahead and just upload your assignment for lecture 6 in that area. Today, before we jump into things, we have a couple of just housekeeping notes here. In the lecture 7 folder, you'll see that we have a lecture 7 data set. We'll have code. The code's actually really light this week, so if you all are a little weary of SAS code, we're light today, so that's okay. We have an assignment data, and then we have the slides, right? So without further ado, let's go ahead and jump right into the lecture. So FNES 702, ANOVA, analysis of variance, right? So last class, what we did was we talked about 3 different types of t-tests, right? Independent sample t-test, or one sample t-test, 2 sample t-test, and dependent t-test, right? So the t-tests were all about comparing groups. One of the really great things about the t-test is that, I think it's really applicable for all that you do, right? It deals with smaller samples, smaller groups. So, my PE teachers here, you have one class and you want to compare it against themselves. You have one class and you want to compare it to another class, or maybe you're a dietitian and what you want to go ahead and do is compare your patients to a national average of sorts, right? And all of these scenarios really work for those t-tests, and I think that that's a really helpful statistical test to know. Today we are going to go over the one-way analysis of variance, also known as the ANOVA test, right? So most folks will refer this test to ANOVA, but really we're going to go over 2 tests today, right? We're going to go over ANOVA and we're going to go over Tukey. So what is ANOVA, right? The one-way analysis of variance, ANOVA, is used to determine whether there are statistically significant differences between the means of 3 or more independent, unrelated groups, right? So what does that mean? So that means, let's say, hypothetically, you want to go ahead and compare the means of 3 different classes, right? So you're a PE teacher and you have your 7 to 8 a.m. class, your 8 to 9 a.m., and your 9 to 10 a.m. PE classes, right? And you want to go ahead and see if there are any BMI differences of such, right? So last week we covered 2 groups, not more or more, sorry, 2 groups or 1 group compared to a national, some sort of standard or some sort of bar. And now what we're going to go ahead and do is we're going to compare and see if there are differences within 3 or more, right? So 3, 4, 5, 6, 7, etc. Go from there. So the second bullet point, the one-way ANOVA compares the means between the groups you are interested in and determines whether any of those means are statistically different from each other. So let's really go ahead and just focus on the second point before we get to the third point, which is this test is going to go ahead and tell you if there are any differences between the groups, right, or the classes, or however, whatever your parameters are here, right? Are there any differences? The next step, what it doesn't tell you is which group is different. And so this is where I said before, this is kind of a 2-part test, is because if there is a difference, then what we do is we conduct a post hoc test or a multiple comparison test called the Tukey post hoc test, right? So let's just take a minute here and let's review. We have multiple groups, right? Group A, Group B, Group C, and what ANOVA is going to go ahead and tell you is A different from B? Is A different from C? Is B different from C? Or is A different from B, right? All of the permutations right here. I might have mixed it up, right? Is A different from B? Is A different from C? And is B different from C, right? Is there any difference from the groups at all? And then let's say the test comes back and says, yes, you know what, there in fact are differences. Then what we do is we run the post hoc test, and then we run, it's called the Tukey test, and Tukey will tell you which group. It might be something like A and C are different, which also means C and A are different, right? Hope this is making sense. But if not, stay tuned. I'm feeling pretty confident that we'll be able to get it, right? So let's give an example of ANOVA, right? So there are 3 separate diets given to 15 patients. Then a test is given where the white blood cell counts are recorded for each patient, right? So now we have 3 groups, and let's just say they're 5, 5, and 5, right? And 5 folks, we give them high protein. Another group, we give them high carbohydrates, and then another group, we give them high fats. And then we go ahead and we measure their white blood cell counts, and we want to see if diet has any effect on this, right? Just giving you guys an example here. I'm not saying that there's any correlation between diet and white blood cell counts, right? And this is just a scenario to kind of help paint the picture. But so the question is to test whether the 3 diets make any difference to the amount of white blood cell count, right? So the way that you set up this model, or the way that you understand this model, is you have a continuous variable, a number, right? And then you have diet, which is nominal, or categories, right? So let's think about this. White blood cell count, that's a continuous number, right? It ranges from 300 to 500, something along those lines, right? And then you have diet, which is diet A, high protein, diet B, high carbohydrates, diet C, high fat, right? Again, a name of a variable. This is also referred to as categorical data, right? And these are the 2 types of data that you need for ANOVA, right? You need a grouping, and you need a number. Now, I'll also make the note that it doesn't have to be diet A, diet B, diet C, right? It could be group 1, group 2, group 3, and in the data it might show up as a continuous number, but you know that those numbers don't mean anything, right? Those numbers are simply labels to indicate which group somebody belongs to. So don't get hung up on that. Don't get caught up. And if you're unclear at the end of the exercise, just shoot me an email, or maybe what we'll do is we'll have a group discussion on this one right over here, okay? And so the question is to find any differences among diet A, diet B, and diet C, right? So this is known as a global test, right? So the ANOVA is a global test. After the global effect is confirmed, further tests are needed to check whether what the differences are, right? Is group A greater than group B or group C? Like I had mentioned before, this test is known as a post hoc multiple comparisons, right? Which will demonstrate in the later sections of this lecture, the Tukey test. Ultimately, if you find that there are no differences, then you don't perform any post hoc analysis, right? You say that you confirm your hypothesis, right? And you say that, look, you accept the null hypothesis and say all the groups are exactly the same. There's no difference, right? So then there's no need to do any more digging if you've already said that all the groups are the same. But if there are different, then you're going to want to go ahead and figure out, well, which group is different, right? Which group is different to isolate the effect. So using this example, maybe you're a researcher and you suspect that, one of these macronutrients are protective for white blood cell counts, and so you conduct this experiment and you figure out, then you find that, hey, look, in fact there is differences. Then you want to go ahead and find out who's the standout, which is the standout group that we're most interested in. Okay, so here we are coming back. It's kind of the same convention, the same flow that we're going to be doing each and every time, right? We're going to write out our hypothesis statement right over here. So the null hypothesis for the one-way ANOVA, it's very similar to the way that we've done them before, right? Except there are multiple groups, right? So you'll see that our HO is U1 is equal to U2, which is equal to U3, et cetera, et cetera, et cetera, for the number of groups that you have, and that is equal to UK, right? U being the group mean, and then K being the number of groups, right? So remember, our null hypothesis is essentially saying all the groups are equal. Group A is equal to group B is equal to group C, right? Or diet A, high protein, our null hypothesis is diet A, high protein is equal to diet B, high carbohydrate, and that's equal to diet C, high fat, right? That's our null hypothesis. So this is first part of the assignment. What is the alternative hypothesis, right? And just a kind of refresher, if the one-way ANOVA returns a statistically significant result, right? So remember, what makes it statistically significant? We set our alpha level, right? We'll set it at 95% or p equals 0.05, and our p-value comes in less than that, then we accept the alternative hypothesis, which is that there are at least two group means that are statistically significantly different from each other. I know that was a mouthful. Pause the video right now and think through the alternative hypothesis if the results are statistically significant. Okay? So take a minute, really digest that and understand that. Remember, the null is that all the groups are equal, and then the alternative, and I almost gave you the answer right over here, but I think you should be able to figure that out. So that's good. Okay, so a note about the ANOVA test, all right? And this is kind of what I've been explaining before and kind of leading up to. The one-way ANOVA is a global test statistic. It covers the overview, right? It's really a high-level test that just goes ahead and says, look, is there a difference or not, right? It cannot tell you which specific groups were statistically different from each other, only that at least two groups were, right? And remember, you can have multiple. You can have up to 6, 8, 9, 10 groups, right? As many groups as you want right over here. And it'll tell you that at least two of them are different. Now, to determine which specific groups differ from each other, you'll need to use a post hoc test. Multiple comparisons, a two-key test. Okay, and like I said before, today's code is really straightforward and really simple, so it's really just going through and kind of reading through and understanding the analysis. Okay, so again, just like all of the ways that we kind of set up our learning, we know what the test does, and now here are the test assumptions. Bear with me one moment. Okay, so every test has its assumptions, right? We talked about this last week. And again, for this lecture, we are treating all of the data as if all of the assumptions have been met. Okay? So, ANOVA assumptions, the first assumption is the dependent variable is normally distributed in each group that is being compared, right? So very similar to t-tests, right? No, technically it's the residuals that need to be normally distributed, but ultimately the results will be the same, right? And let's come back, and if you think back to how we calculate variance and such, and thinking back to kind of some of the previous lectures, this should make a lot of sense. There is homogeneity of the variances, right? Meaning that the population variances should be equal or very close to equal, right? Again, this is pointing at, we don't want big outliers in the data, right? We don't want to have the data being very skewed. We don't want to have it skewed in one direction. All of this should be normally distributed. These are the assumptions that we are making, right? and then an independence of observations, right? And this is, let's go through this. A common assumption across all inferential test statistics is that the observation in your sample are independent from each other, meaning that measurements for each sample are no way influenced by or related to other measurements of other subjects, right? So going back to kind of our diet example, right over here, the folks who are having high protein, they're not having high protein and high carbohydrate, right? They're not having high protein and high fat. The high protein group is the high protein group. There's no cross talks between the other ones right over here. This is really like the high protein group can't end up in the high carbohydrate group and such. There's independence, right? These are three very separate groups that we're talking about here. All right. Okay, and then so just like the t-test had a t-statistic, right? The ANOVA has what's called an f-statistic, and the f-statistic is the ratio of the mean squares to the mean squares error. So think about that for 1 second, and I'm going to Okay, I'm back. Sorry about that. So really, ultimately, at the end of it, the F statistic is calculated by taking the mean squares and then treatment divided by the mean squares error, right? And I promised you all that we're not going to actually calculate any statistics. So the main point here is this. The larger the F statistic, the greater the variance between sample means relative to the variation within the samples, right? And look, when there's a lot of variance, what does that mean, right? When there's a lot of variance, that means there's going to be a lot of standard deviation, right? Like the standard deviation number is going to be higher with the variance number. Does that make sense, right? So the key takeaway is the larger the F statistic, the greater the evidence that there is a difference between group means. So that's really the main point that you want to go ahead and think through here, right? The larger the F statistic, the greater evidence that there is differences between the groups. Okay, so we're going to go ahead now that you know all of this, we're going to use and run through an example, right? And it's the same example that we just did, that we went through earlier. 3 separate diets are given to 15 patients. Then a test is given whether the amount of white blood cells, where the amount of white blood cells are recorded for each patient, right? So again, we have 3 groups. One group is eating a high protein diet, the other group is eating a high carbohydrate diet, and then the last group is eating a high fat diet. The question is to test whether the 3 diets make any differences to the amount of white blood cells, right? Oops, I got a little cut and paste error here. Sorry, everyone. You can just go ahead and delete that from your slides. My bad. Okay, so using the diet.xlsx data, we're going to state our null and alternative hypothesis. We're going to create a box and whiskers, right, displaying each diet. And what this is going to do is this is really going to help you kind of visualize and see if there's a difference. We're going to calculate the F and P value, F value and P value, which SAS is going to do. And then we're going to interpret the results. And then we want to know which group are different, right? Okay. So now I'm going to toggle over to SAS. And this is the ANOVA code that is uploaded on Blackboard. And you'll want to upload, right-click, upload files, and upload the code. And you'll want to upload the data set, which is called 2-sample t-test. Oh no, I'm sorry, called diet. And then here we go, right? So I'm going to name this temporary data set ANOVA for this exercise, right? I'm going to right-click on my diet file, hit Properties, and you'll see that we'll have the file path here, which we'll replace here. And then we'll add in our sheet name, which happens to be sheet 1, and we're going to do our running man. So you'll go ahead and you'll see we have essentially two columns, right? We have our A1C column, which is the white blood. I don't know why it's called A1C. It should be white blood count. But regardless, this is A1C should be white blood count, and then diet A, diet B, and then diet C. Okay? It's just a column name. And then what we're going to do is we are going to run our proc ANOVA, which is another statement now, right? Remember, you're going to have lots of different ones. You now have proc means, proc univariate, proc t-test, right? These are all different types of codes that you're able to go ahead and run. So let's go over this. We're going to say we want data equals ANOVA. Our class or the group that we're interested in is diet, right? And if we look back at our results, this is which diet they're having, right? A is high protein, B is high carbohydrates, C is high fat. And then our variable of interest is the A1C column, which really means white blood cells, right? Equals to diet. And then we want the means of the diet. We want to break it out by group. We want the 2 t-test. And then this CL diff right over here, which is essentially going to go ahead and take the results of the 2 key and tell us which one the groups are different from. Okay? So let's go ahead. Highlight. Hit the running man. And now you're going to go ahead and get our results. Okay? And you'll see number of observations read, 15. Diet has 3 levels, right? And the levels are diet A, diet B, and diet C, right over here, right? So that's how you go ahead and you interpret this one. You'll see that our model has 2 degrees of freedom. Degrees of freedom is always calculated by levels minus 1, okay? So the main thing that we're going to go ahead and be interested here is our F value, which is 16.78, and then our P value, which is 0.0003, right? Which tells you now that our model is, I'm going to pause, let you answer, and the answer should be the model is statistically significant, that there are differences between diets A, diet B, and diet C, right? So now we're saying, oh, okay, so there is a difference, right? One of these diets or 2 of these diets may differ from each other and result in different white blood cell count numbers, okay? So let's go ahead now, and I had asked about a box and whiskers, right? So you'll see that the ANOVA essentially does it for you because it's actually really important. So let me zoom out a little bit here. You see that we have now a figure. This is our box and whiskers, and this is our white blood cell numbers. And you see, look at that, right? Diet A, the median is close to 800, whereas diet B, we're probably at, what is this, like 560? And then diet C is pretty close too, and that's at like maybe 540, 550, right? And the median, and then look at the kind of the outliers, right? And you have your one outlier here. Remember, each outlier is roughly about 2 standard deviations away, right? So here's the min, the max, these are our outliers. And look, even the minimum for diet A, the high protein diet, is higher than diet B and diet C, right? So our model says that, look, ANOVA tells us, look, there is definitely a difference between these 3 groups right over here. Now, take a moment, right, and think what groups are different, right? Pause, think, what groups are different? Okay, now, hopefully you paused and you thought about it, but group A is different from group B. Group A is also different from group C, right? Now, it's possible, oh, and then also just on that note, right? So the inverse is also correct. Group B is also different from group A, and group C is also different from group A, right? But is B different from C, or is C different from B, right? We don't know. That's why we have to perform the what test. Pause and answer, right? What test? What type of test do we have to now go ahead and perform? The answer should be a post hoc test, right? We're going to perform a post hoc test, and we're going to perform the Tukey post hoc test, which we automatically put in anyway. So, you know, for the sake of time, I just left this code in, but normally you wouldn't run ANOVA with this. You'd find that there's a difference first and then throw in the Tukey, right? But it really doesn't matter. You can put it in either way. My suggestion is put it in, see if the model is statistically significant or not. If it's not statistically significant, then just stop right there. Don't read the Tukey results. But since it is statistically significant, we're going to go ahead and we're going to roll in. Let me zoom in a little bit more. Gave it to us twice. And now you'll see, look, our alpha was set at 0.05, right? And it is statistically significant. Therefore, you go back and you look, and anything that has these 3 stars right over here is going to go, and these are going to be the values that are different, right? So you'll see diet comparison. A is different from C. Yes, it is. A is different from B. Yes, it is. C is different from A. Yes, it is. We skip one. B is different from A. Yes, it is, right? Now over here, this is saying C is different from B. No, it's not. And then B is different from C. No, it's not, right? And if you go back and you look at this figure, you say, oh, okay, right? Look at that. So if you all are inclined, it would be great for you to go ahead and take the B and C data and then run a what type of test to see if there's differences between these 2 groups only, right? Something to go ahead and think about, right? So this is not, there's no assignment here, but how would you do that if you were to go ahead and do this one, right? So ultimately, the key takeaway is, if the model is statistically significant, you come down and you read the Tukey results, and the Tukey results will tell you these are the groups that are different from each other. A is different from C, A is different from B, and B is different from A. Okay, good. So let's come back. So our answers right over here, right? Hypothesis statement, all the groups are equal. Our alternative is at least one group's mean is different from the rest, right? Here's your box and whiskers. This is your F value, 16.78, which you find next to the P value, which is 0.003, right? And how we can interpret this, and start the interpretation at least, is since the P value from the ANOVA table, 0.003 is less than the alpha at 0.05, we reject the null hypothesis, right? So let's come back. What was our null hypothesis? That all the group means are equal. Well, it's not, because it's different, and it's statistically significant. The differences are statistically significant, right? So, right, so at a significant level of 0.05, the conclusion is that the typo here, the diet methods make a difference on the impact of the result, okay? Now, since they are different, which groups are different? And what we would say is under the Tukey's standardized range, HSD, test for A1C, to tell which group have means that are different, we must look at the pairwise comparisons that have stars next to them. From the table, we can see that the mean values for group A and C and A and B are statistically different, okay? So, when you have this on an assignment, when you have this on the exam, this is how I need you to start leveraging the language, right? These here, since the p-value, this bullet right over here, this bullet from right over here, this is how you need to start going ahead and utilizing the language when you go ahead and report it out. And this is going to go very, very far away for you, especially when you start taking the research method class, right? So it's good to start getting into the habit of speaking this way, right? So ultimately, the key point is here. If there are 3 stars next to it, these are the groups that are different from each other, right? So how do we then go ahead and interpret the results, right? We would say this. A one-way ANOVA was performed to compare the effects of 3 different diets on white blood cells. A one-way ANOVA revealed that there was a statistically significant difference in mean white blood cells between at least 2 groups, the F equals 16.78 and the p-value at 0.0003. Notice I just made some edits here, some quick typing. There's some typos, so fix your slides for your records, right? Tukey's HSD for multiple comparisons found that the mean value of exam scores were significantly different between diet A and diet C, as well as diet A and diet B. There was no statistically significant difference in mean exam scores between method C and method B, right? So this is how you have to go ahead and kind of frame your answers and frame your results, right? And you would just essentially plop in your numbers here and then find out which groups are actually different from your perspective, all right? So, lastly, for your assignment today, what we're going to do is we have a PE class, right? And 4 separate exercises are given to 20 students. So we split the 20 students up, and we want to go ahead and see, hey, these are some different types of exercises that we're going to do, and we're going to see if that has an effect on the number of push-ups that they can complete in a period of time, right? So group 1 is only going to have body weight training. Group 2 is only going to have resistance training. Group 3 is body weight and resistance training, and then Group 4 gets no exercise at all. They just get to play basketball, right? So the question is to test whether the 4 exercises make any difference in the amount of push-ups. And then let's delete this. You know, I'm gonna repost these slides, so don't worry about making those corrections. But using the ANOVA homework.xls, state your null and alternative hypothesis. Calculate a box and whiskers, displaying each training group. Calculate the F and P value, interpret the results, which groups are different, right? And I want you to use the language that I set forth in this slide. Okay, that's it for today, folks. I hope that this is good. I think that this assignment should keep you busy for a little bit. I really recommend you watching this one more time and reach out if you have any questions. Thank you.